So thank you to we had excellent presentation, great overview on tough topics. Start our discussion now. To all attendees, remember there is question and answer or a chat button. You can put your questions through those buttons. I am Laurent Zeta. I am so pleased to moderate together with my good friend Rufino session with these very renowned speakers. Christina, you gave great on results from different trials. A couple of days ago, something new on the usage of a 8 milligram has been released. Can you give us your ideas on that?
Sure, Dr. Lanzetta. Well, it's really hot off the press. I think it's around 24 hours ago that the Pulsar results were released for Regeneron and Bayer's 8 milligram high dose of aflibercept. So we all are familiar around the world with aflibercept, which has been widely used with a great safety track record for over a decade now. And the impetus behind Pulsar was that perhaps a higher dose of aflibercept, specifically 8 milligrams, which is 4 times the standard dose of 2 milligrams, might offer longer durability for wet AMD patients. And because of the encouraging signals seen in the phase 2 Candela, there were 2 pivotal trials that were conducted, Pulsar and Photon. Pulsar is the one for wet AMD, Photon is the one for DME. And Pulsar is a double-masked, global, multi-center, non-inferiority trial. It compared aflibercept 2 milligrams, which is the standard dose, every 8 weeks versus a high dose of aflibercept every 12 or every 16 weeks. And of note, the patients had a chance to drop down to Q8-week dosing if there were certain pre-specified criteria that were met. And they looked at the primary outcome of visual acuity, which was at week 48. And pretty impressive results in terms of the durability. Almost 80% of the wet AMD patients were actually maintained on either Q12 or Q16 week dosing intervals through week 48 without any new safety signals seen. There's also a higher percentage of patients in the high dose aflibercept group that did not have fluid in the central subfield at 16 weeks, which was one of the pre-specified secondary endpoints. And so they're planning to submit this data to the regulatory authorities in the coming months worldwide. And so we may have yet another option to offer our patients in this quest for durability that we're all on right now. So pretty exciting.
Christina, thank you, Christina. I have one question for you. Do we have room for PDS, consider the long-term effect of these new drugs?
I definitely think so. I think, PDS really reaches to a new level when it comes to durability. These longer acting agents we're looking at are looking at around Q16 weeks, Q20 weeks, maybe up to, with the TKI, 6 to 9 months. But with the port delivery system, that was looked at for every 6-month refill exchanges in their phase 3 and perhaps even longer suggested by the phase 2 ladder study, I do think there's room for that. And if you look at some of the patient experience studies as well, patients seem to be very satisfied with that. So someone who's a good surgical candidate who requires very frequent therapy may indeed be a great candidate to receive PDS.
Thank you so much, Christina. I think we move to Hendrik and Arshad. I to ask a question to Hendrik. Thank you so much, because this was a great summary on Stargardt disease, and we are very grateful for this overview. So you mentioned that gene therapy is another possibility. What is today difficult with the current technology? gene therapy?
I mean, it's an obvious therapy for a monogenic disease that, by the way, is not that rare. I mean, we all know that, especially people that see geographic atrophy in their clinics, that Stargardt disease is not that rare. So it's unfortunately a large gene. So the standard approach that we heard of by the approval of Luxturna would not work because the packing capacity of these carriers that we call AAV is too small. So you need a technology to bypass that problem. Therefore, there are more elegant approaches, such as gene editing approaches, specifically base editing that I presented in my presentation, that may be superior. In the case of gene editing, you do not actually provide the whole gene, and you really repair the actual mutation in the existing gene, which should come with superior safety and efficacy, but it remains to be determined because we are still in the preclinical phase. But certainly, gene editing technology is a preferred approach to target Stargardt disease.
Hendrik, we have 3 more minutes, so Rufino, please, it's on you with some questions. Rufino, you're on mute.
So this is a question. Thank you, Paul. This is a question for Professor Achad. The question is related with the outcome we have on the evaluation of geographic atrophy and the progression and with treatments. So this is a final outcome, a very final-oriented outcomes. Do you suggest that in future studies, other outcomes could be evaluated as efficacy outcomes for new drugs? Because previous, for example, using OCT, you suggest any kind of different outcome?
Thank you. Great question. I want to thank you and Dr. Lanzetta for having me here. I think the difficulty, obviously, we have with geographic atrophy is that, we need an outcome that we can measure in first year or second year. We cannot do a 5 or 10 year study. So obviously, the primary outcome in the study is mean rate of growth of GA, and it's looked at in different ways. Square root or non-square root transformation, also the slope that we saw with the Zymura trials, the GATHER2 study. So I think the primary endpoint is accepted by the FDA, so I don't expect this to change for future drugs. But obviously there are other things we're still learning about: functional outcomes, low luminance visual acuity, reading speed, microperimetry. And again, those are very hard parameters because there is a subjective impact on those parameters based on patients' effort and other things. So I think as a field, we continue to look for more biomarkers that we can evaluate to see a treatment effect.
I would like to pose audience which came in. It's not directly related to your talk. To intervene, obviously. This question regards Faris. The question is whether compare PDR patients to non-proliferative diabetic retinopathy patients, and the asker who's well, I cannot read because of presbyopia, and he or she also want to know in what this may happen, and if this may be useful in the management of patient with PDR.
So very quickly, because we're just about running out of time. So this has been $1 million question with all anti-VEGFs, and a lot of the time the retina seemingly looks better in patients with diabetes, but then if they don't come back, or if have a stroke, or they go on dialysis, or they have their legs amputated and they get lost in the system, a lot of them come back with rhabiotic glaucoma or very severe proliferative diabetic retinopathy. So really our current state at the moment is that when you need to do the laser, you do the laser as well as manage some patients potentially for diabetic macular edema, and or you might be able to help yourself not to have such big vitreous hemorrhages with some anti-VEGF. But for long-term management of PDR, laser still has its place.
Thank you very much, Tunde. Thank you for the next presentation, and I close of this very interesting session.
Thank you very much. So, thank you also to the audience. These were excellent presentations, and we have a great discussion. Thank you. I wish you a great meeting.
Thank you, everyone.
Thanks, everyone. Bye.
Thanks.